everyone, and welcome to today's live broadcast, Found in Translation, ST2 from Discovery to Clinical Adoption, presented by Dr. James Snider. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events, and webinars advancing scientific collaboration and learning. For more information, please go to labroots.com. I'm Christina Mahalik. I'll be your LabRoots moderator today. Before we begin, I would like to remind everyone that this is an event and it is interactive. We encourage you to submit questions as you think of them. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right corner. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now I'd like to introduce Dr. James Snider. Dr. Snider has been the president of the Critical Diagnostics Incorporated since August 2005 and serves as its member of the management board. Prior to joining CCD in August 2005, Dr. Snider served as an executive vice president of business and operations of Intelligent Medical Devices Incorporated. Dr. Snider joined Intelligent ND in October 2001 as Vice President of Marketing and Business Development. Prior to IMD, Dr. Snider served as a series of marketing and product development positions at Applied Biosystems, also known as ABI. In his last role at ABI, he managed the sample preparation systems product line and team from genesis through product launch and successfully established it in the market. Before his 8-year tenure at AB, Dr. Snider served as senior scientist at Biotech Research Laboratories, a contracts and service research company specializing in retroviral technologies. He performed his postdoctoral research as a fellow in the Biological Response Modifiers program at the Frederick Cancer Research and Development Center at the National Cancer Institute. Dr. Snider holds a BS in chemistry from Grand Valley State University, a PhD in chemistry from the University of South Carolina, and an MBA in technology management from the University of Phoenix. Please join me in welcoming Dr. James Snider. I will now turn the presentation over to him.
Okay, thank you, Christina. Appreciate that kind introduction. Welcome, everybody. It's certainly my pleasure to have an opportunity to speak with you today and to present this material. What I've prepared for presentation today really is, I guess, better described as a case study in taking a biomarker from the discovery phase all the way through clinical implementation. So throughout the presentation today, I'm going to be interspersing some very technical clinical detail, but that's not used necessarily to make you experts in the field that we work with this biomarker. It's really intended to give you perspective on the analytical, clinical, and technical requirements and expectations around a biomarker development program. So in addition to the clinical and technical detail, I'll be also discussing some of the business and regulatory elements as well. And in none of the instances am I going to go into great detail because this is not intended to be a true description about how, for instance, how to get a regulatory approval for a test, because each product is unique. But it will give, I'm hoping, more of a comprehensive representation of the entire scope from, again, from discovery to implementation. So why don't we proceed with the material? So this slide basically summarizes, a biomarker assay development program. So Critical Care Diagnostics, as you notice from the title, has focused on the biomarker called ST2. ST2 is used in cardiovascular disease, particularly in heart failure. And these are some of the questions that you would need to address as you're considering or adopting a biomarker assay development program. So first, is the analyte involved in a disease? So there are many ways to address that. In the case of ST2, it was found in translation, which means that the investigators that first discovered that ST2 was involved in cardiovascular disease had a very unique cellular model that emulated a certain form of cardiac disease, specifically heart failure. And then they looked at which genes were upregulated in that model. And surprisingly, ST2 was upregulated. Prior to that, ST2 was known to be involved in other diseases, but that novel discovery that it was involved in cardiac disease and in heart failure was unique, and that was the discovery that was the foundation for the intellectual property that led to the formation of our company. And then you take that a step further. So it was involved in disease. What is the function of the analyte in a disease? That's a much harder question to answer. And I'm going to touch on some of the clinical analyses that we did to get a better understanding of that question. What reagents exist to measure the analyte? Is there a way to measure it? As I just mentioned, the original discovery was from gene expression analysis. So obviously that was nucleic acid techniques, principally PCR and northern blot analysis. But in the clinical laboratory, those are not widely used technologies for routine use. So you need protein-based assays for blood tests or for other biological fluids. So those are typically antibodies. So do those exist, or do they need to be produced? Very importantly, what are the analytical requirements for measuring this analyte? Is it stable? Is it unstable? What biological fluid is it present in? Is it a biological fluid that is easy to obtain, or something that would be very difficult to obtain? Is it a high concentration analyte or a low concentration analyte? For instance, you're probably familiar with tests for cardiac conditions like a myocardial infarction. And in the laboratory, a very typical test for an MI is troponin. Troponin is a very low concentration analyte, so those tests have to be very sensitive. Other tests have much higher concentrations of their analyte and don't require that sensitivity. So it's really important that you have a knowledge about the specific clinical context and the requirements for how the analyte is going to be used. And then are those reagents available to meet that requirement? So if you have antibodies, are they good antibodies? Do they have the right affinity? Do they have the right sensitivity and specificity? All things that have to be understood and characterized. The form factor. Is this a test that needs to be done very rapidly? I mentioned troponin for myocardial infarction. In MI, a myocardial infarction or a heart attack needs to be treated quickly. So you want an answer to whether the patient who has chest pain is truly having a myocardial infarction as rapidly as possible, certainly within an hour, so that you can begin the appropriate therapy. So a test that would take 4 hours or 5 hours to complete would not be an appropriate solution for that problem. However, a test that is not used for disease diagnosis may not require such a rapid turnaround, and consequently a technique that might give you better analytical performance but takes more time might be a better solution. And those things all need to be considered. And I'll discuss that a little bit in our experience. And then what is the business case? Is there a true need for the test? So is it disease diagnosis? Is it establishing a prognosis and a risk profile for the patient going forward? Is it a companion diagnostic? And this is very common in the oncology world, where the therapies are very, very expensive and very specialized to unique clinical conditions. So there will be a test that's specific for that condition that would then define whether that therapy would be implemented. That doesn't exist in cardiology, but we still have the idea of a complementary diagnostic routine, where can we use tests to evaluate whether patients are responding to the therapy that they're receiving, even though therapies are much more generic. Are the patients responding to these therapies, or should we consider something else? So it really isn't the companion diagnostic, which would define whether a patient should or shouldn't get a specific therapy, but whether the patient is generally responding to therapy or not. So all of those things have to be understood in order to even determine whether your program is worthwhile going forward, how you're going to go forward with it, what investment should be made in the program, what your possible return and future opportunities are. So this is a quick snapshot of where we are from beginning to current with the evolution of critical diagnostics. So as Christina said in the cover, the company was formed in 2004. That was based on intellectual property patents that came out of Harvard University. And that was around the use of ST2 in cardiovascular disease. And then we actually started operation of the company in 2005 when I joined. And from 2005 until 2008, we worked to develop our own proprietary set of reagents based on experiences that we learned from some what are called research use only, or RUO reagents. So reagents that are available from various biotech companies, but not necessarily validated and not necessarily used or even appropriate for use in a clinical context, but can certainly be used in some instances to understand the biology and maybe the clinical context of the analyte that you're interested in. In 2010, we completed our development of our proprietary assay system and had obtained a definition of a clinical use, and then we moved that into the regulatory process, obtaining our US FDA clearance for the test for our first generation test, which was an immunoassay based on an ELISA format, so an enzyme-linked immunoassay system, which is a fairly cumbersome multi-hour test that is usually involved, immunoassay system that uses antibodies. In 2012, we began to expand that from our US launch into an international sales effort, and also began development of a second generation product, which was based on lateral flow technology. So very similar to the home pregnancy test type of technology, a little bit more sophisticated in that it's quantitative and uses an instrument reader, but a rapid test that could emulate the performance of the first generation test. Now, other important elements are, once you have a test, getting FDA clearance or regulatory clearance in whatever country that you're trying to do business in is necessary. That's the first step in being able to legitimately sell a product for clinical use. But then there are many other steps that need to be achieved. Inclusion in the guidelines. So every medical field has a professional guideline or a professional organization. In cardiovascular disease, it's the American Heart Association, or the AHA, or the American College of Cardiology, the ACC. They combine to create guidelines for every form of cardiovascular disease. And in 2013, ST2 was first included in the AHA-ACC guidelines for heart failure management. So that was a major milestone of recognition that the test has clinical value and was being recognized by the professional associations in the field in which we produced the test. That was followed up by other requirements in order for the test to be used in the United States. One is to have an analyte-specific code for products. All products have to have a code. The analyte-specific code is specific to laboratory products, and those are defined and issued by the American Medical Association. There are very specific expectations and requirements in order for the AMA to issue such a code. We were able to achieve that in 2014. And then once a code is issued, CMS will make a payment decision. Center for Medicare and Medicaid will make a payment decision based on those codes. It may be favorable to the manufacturer, it may not. We went through the process of petitioning for what we felt was a fair and favorable payment decision, and we achieved that. So early 2014, we received the analyte specific code. Late in 2014, we received a favorable payment decision by CMS. In 2015, we obtained our CE mark. So CE mark is the European standard for regulation of clinical products. For our lateral flow product, we obtained that mark and clearance in 2015, began marketing in Europe in 2016, and also then used that CE mark standard to begin market introduction in our Asian partners. And we currently do business in about 40 countries around the world. So the US, all of Europe under the CE mark standard, and in most Asian countries with the exception of Japan. So that gives you a snapshot of, basically, 11 years from the time in 2005 when we truly initiated the project until 2016, where we have 2 products on the market and doing business in multiple countries. Okay, so let me talk just a little bit about some of the technical details to give you some perspective on the challenges and requirements in understanding, defining what it is that you're trying to do, and taking it through these various steps. So first, and this is just used for illustration of the complexity of what some of these biomarkers can represent. So ST2 is one of the proteins in the interleukin-1 receptor superfamily. It exists in 2 forms, as a membrane-bound form and a soluble form, and our test measures the soluble form. It's been known in the literature and in science for many, many years, since 1989, because of its involvement in the autoimmune disease states and part of the T-cell modulation process. So as I'd mentioned, the discovery by the investigators at Harvard that ST2 was also directly involved in cardiovascular disease as part of a cardioparacrine signaling mechanism was novel and unique, and that's the discovery that we leveraged moving forward. This cartoon more clearly represents that, where we can see that the membrane-bound form, soluble ST2L, exists on the cardiomyocyte membrane. The cardiofibroblast, which is the second but more predominant cell type in the heart, expresses both ST2 as well as interleukin-33. In the context of a diseased heart or a stressed cardiomyocyte, the cardiomyocyte is looking for a protective signal through interleukin-33, which is expressed by the cardiofibroblast. So interleukin-33 would bind to the receptor form of ST2. The myocyte then goes into a recovery mode and responds to therapy, and typically will overcome the stress or the disease that it's exposed to. However, let me close out of this. Oops. In some individuals, an alternate pathway is initiated, and in this case, the cardiofibroblast may also upregulate the expression of the soluble form, which in that case, it binds interleukin-33 so that it's no longer biologically available to the receptor. In that case, now that stimulated myocyte does not get the protective signal from interleukin-33, and the myocyte then transitions into an alternate response pathway of apoptosis leading to necrosis, fibrosis in the tissue, and worsening disease. And that's exactly the clinical response that we see when patients have an elevated level of soluble ST2 and the presence of cardiovascular disease or symptoms of cardiovascular disease. So as we were trying to understand this in clinical context, our very first clinical trial, which was a trial of patients, and as you can see in this case, about 600 patients, that presented to the emergency department of Massachusetts General Hospital with symptoms of heart failure. So dyspnea is a formal phrase for shortness of breath. So these are patients that have congestions in their lungs, they've got pressure in their chest, they're having trouble breathing, they typically cannot walk very far. and they may even have difficulty breathing just standing. So they present to the hospital. Most of them have a history of heart failure, but they may also have other diseases that can result in very similar symptoms. So other diseases that can create similar symptoms would include pneumonia, which is a very common co-presentation, an asthma acute exacerbation, a COPD, chronic obstructive pulmonary disease exacerbation, even a myocardial infarction may, but that's much more rare. So in these 600 patients, just over 200 were adjudicated diagnosis of acute heart failure. So that was, of course, the syndrome that we were interested in understanding better. And the other nearly 400 patients had other symptoms, other syndromes, resulting in their shortness of breath. Now, when we looked at this, we do see, now this is using a research use test, so it turns out that we didn't know it at the time, but there were some analytical issues, some performance characteristics that were not ideal with those research use reagents, but we needed to use them to get a better understanding of the clinical context and the clinical value of the biomarker. So in this, what we're looking at in this box whisker plot is the differential signal or concentrations of ST2 in those patients that have symptoms. So they all have they're all sick with something. Again, about 200 of them were diagnosed with acute heart failure, and their ST2 concentrations are higher than those patients who had symptoms but did not have acute heart failure. So just at first pass, this would look like there is a diagnostic differentiation. It's not absolute. Of course, the perfect diagnostic test would be positive in patients who have the disease and negative in patients who do not have the disease. Very few diagnostic tests meet that criteria. There's usually some overlap of higher concentrations in disease and lower concentrations in the absence of disease, more similar to what we see here. Now, if there was no other test for diagnosis of acute heart failure, this may be good, but there is a test. There's a test based around natriuretic peptides. There's two different forms, BNP and NT-proBNP. That's not important for this discussion, but those tests actually work really well for diagnosis of acute heart failure. So our first pass, when we looked at this, there's a signal, but it's not better than an existing test. So when you think about your business case, there would not be probably reasonable justification to pursue ST2 as a heart failure diagnostic test because it's not superior to an existing well-established product. But what we saw is that not only was ST2 concentrations elevated in the presence of disease, but they also correlated with the severity of symptoms. So the New York Heart Association class is a representation of the symptom severity. So there's four steps. Very few patients present to the emergency department with the lowest level of symptoms, which is category 1. Most present with symptoms at the categories level 2 through 4, and the most severe is level 4. So those are the sickest patients symptomatically. There's another scale, A through D, that represent the underlying pathophysiology of the disease or the stage of the disease. So in most cases, you have to take both the scale and the New York Heart Association class symptom representation to get a real reflection. But symptom presentation is important. So ST2 is higher in patients who are sicker, so that's important. And then surprisingly, and this was the observation that we ended up recognizing as the unique clinical and potentially the business value proposition with ST2, was its very powerful prognostic value. So this is an analysis that's called Kaplanmeier, Kaplanmeier survival. And what we're looking at here is a ST2 concentration at a specific concentration, and then whether patients that were above or below that concentration, what their mortality events were over a period of time, in this case for 1 year. So the solid bar shows that below a certain concentration, only one patient died in 1 year, and that patient was after 6 months. So it shows that patients, even though they're sick, they're presenting with disease. In this case, we're only looking at the patients with acute heart failure. So they were sick enough to go to the emergency room, they already had underlying disease, but if their ST2 concentration was low and they received therapy, their risk of mortality over the year was very low. Again, only one patient died. By contrast, if their ST2 concentration was modestly elevated or maybe even significantly elevated, now they had a much, much higher risk of mortality, and in many cases very rapidly. And it turned out that ST2 is a very powerful predictor of short-term risk within 30 days, which is a time frame that is actionable for doctors that are seeing these patients. That's in the time frame where they can make changes to therapy, monitor the patients, and then assess going forward. 1 year mortality or be longer is much, much more difficult to manage clinically. So this discovery then gave us a differentiation. We didn't have a differentiation based on diagnosis of disease, but we had a very clear and powerful differentiation of this marker from existing tests or existing parameters in evaluating heart failure patients to establish a tool for prognosis. Now, prognosis, though, of course, is only useful if you can do something about it. Having a patient come in and saying, well, you've got a 3-fold higher chance of dying within 1 year, thank you for coming, is not an effective, clinical management technique. We have to be able to do something about it. And as we get further into the presentation, we'll describe what we've discovered about doing that. But one of the things that we had to do was also evaluate what reagents we were going to use going forward. So once we established that there was clinical value in having an ST2 test, then we had to evaluate whether we would use some of the existing reagents or whether we would need to develop our own, and whether the existing reagents had the right performance. Okay? So first I'm just going to put this reference out. This was published several years ago, back in 2012, by Nadir Rafi. Nadir, at the time, was the editor-in-chief of the journal Clinical Chemistry, which published this. Nadir is also the head of the laboratory at Brigham and Women's Hospital in Boston. And what he pointed out in this editorial article is that investigators can be significantly misled, and the field can be misled by using research use reagents if there's a characterized and validated test available. The difference is that many of the research use reagents might be perfectly functional. They may work well, but those producers, those manufacturers, usually only go through very modest validation and standardization protocols, and consequently, you may not understand or recognize that those tests could be affected by inhibitors that naturally occur in your biological specimens. They may not be as specific to the analyte that you're interested in. They may be compromised by sensitivity. And that was one of the things that we learned as we were moving forward, is that the research use reagents that we were using, because they had not been fully characterized and were not validated, did not provide the true performance that was necessary for a clinically used test. So we had to go through a program to develop our own reagents. These kind of protocols are fairly routine. There's many techniques and service companies that can do production of an antigen, of a recombinant protein to use as an antigen to raise antibodies, companies that can then take that antigen and make antibodies, companies that can characterize the antibodies, and so on. So we obtained, or sourced the projects to those companies, developed our reagents, and then we had to determine what format we wanted to use. ELISA is the generic, more standard first-generation format. It's very easy to do. It's not a fast test. It typically takes 4 to 6 hours to perform an ELISA test, but you can do multiple specimens per plate. It's a 96-well plate typically. Colorimetric or luminescent technologies are the normal options. Colorimetric is particularly easy. It's widespread. It doesn't require any unique instrument systems, no equipment, but it's not as it has relatively modest sensitivity. Luminescence is a much more sensitive, much more precise technique, but is more specialized equipment. Lateral flow is also a reasonable technique. It's widely used. It can be adapted quickly, but it has some performance characteristics that may not be completely suitable to initial characterization of a test. And some of those compromises are absolute sensitivity. It usually requires fairly high concentrations of an analyte to accurately measure. And it's a relatively noisy technology, so getting good precision is a challenge with lateral flow. It's not impossible, but it's not easy. Of course, the major companies, the Abbotts, Roches, and so on of the world, have the homogeneous automated assays, which are very, very nice. But those programs can take multiple years. A typical development program from discovery and identification of your reagents to validation may take 2 to 3 years for those large instruments, integrated instrument systems. And if you're trying to understand what your biomarker is and what your clinical context is, that may be too long a period of time to do assay development before you begin doing your clinical validation. So ELISA is a typical technique that's used, and that's the technique that we used. Now, when we were evaluating our reagents, and we picked our reagents, one was for performance, but also for control. If we were going to build a test, we had to make sure that the reagents that were going into our test, because we are the manufacturer of record, we had to know that the quality of those materials was consistent over time, and that we could defend the production of this test to go into the market. So rather than relying on a third-party source of these reagents that we didn't have high confidence in anyway, we produced our own. And in some of our first analyses, we discovered that the performance was profoundly different. And we saw two things here. And I don't expect you to understand all the details in this table, but if we look at the columns of the MBL, which was the research use reagents that we used, and CCD, which is our reagents, there's a huge difference in the numbers. So it turns out that our test was about 60 to 100 times more sensitive than the MBL reagents. At first, I wasn't sure, whether we were measuring, other materials. Maybe the two tests were not actually measuring the same analyte. With further investigation, we learned that in fact we were measuring the same analyte. But again, one of the things that is a point of emphasis, if you're in a biomarker discovery development program, is understanding your analyte. In this instance, we made the recombinant protein, which was used as the antigen for our antibodies, we did a human expression system. So the antigen that was used to make our antibodies was as close to the native protein as we could possibly get in a recombinant system. The research use tests used different expression systems that were eukaryotic but were not human. And because this is a highly post-translationally modified protein, the antigenicity is different, the sensitivity and the affinity of the antibodies is different, and consequently you get very different performance of your test. So our test that was made to the human characteristics of the protein had, again, much, much higher sensitivity and was not as sensitive to certain inhibitors. So in this case, if we compare EDTA plasma to heparin plasma, we see that heparin completely obliterated the sensitivity of the research use test, but had no effect on our test. And that's very important because in the field of cardiovascular disease, patients are very frequently treated with heparin as an anticoagulant. And in further analysis, we discovered that the MBL test was so sensitive to the inhibitor that even to heparin, that even therapeutic concentrations of heparin affected the test. So those kind of things can compromise your clinical interpretation and need to be understood. So this was an observation that we made early on and further gave us further justification that using our own reagents was going to give us both a business advantage as well as a clinical performance advantage as we moved into our validation. Interestingly, though, if we look at a method comparison technique, and this is a standard statistical method comparison technique called passing the Bablock, where we're simply looking at whether the two tests, give you comparable interpretations, and in fact they do. So regardless of which test you're using, if an ST2 concentration is elevated, it's elevated in both of them. The absolute number is going to be different, but high is high and low is low, and they compare in that kind of a crude comparison. Another comparison technique, and this is also very standard in the diagnostics at test development and use protocols. This is called receiver operator curve analysis. And what we're looking at is a reflection of a test's sensitivity and specificity for a specific question. In this question, we were looking at mortality in 1 year. So whether patients survived or did not survive over 1 year. Now, the perfect test would have an area under the curve, which is, there's the area to the right of the curve, of 1. So you want this curve to be perfectly vertical on the y-axis and perfectly horizontal across the top on the x-axis, and that would give you an area of 1 under the curve. In this case, we see that our area under the curve is 0.8, just a little bit above 0.8 for each assay. So again, if ST2 is elevated, you get an accurate representation of a risk of, in this case, an outcome of death, and both tests can do that. But to the next slide, we see that the optimal concentration is about 100-fold different between the two tests. Our test measures at a calibrated value of 27 nanograms per mil and only 0.22 nanograms per mil for the other test. And again, these two tests are not, you cannot compare numbers between these two tests because they are not calibrated the same. There is no gold standard calibration for ST2 as an analyte. That was something that we had to do as well. So we see that the biomarker is consistent. What changes is how accurately and how consistently you can measure it based on the two tests. Now, as we move forward, and this is one of the requirements for definition of a biomarker and of any other clinical laboratory test, and that is whether you can measure it in normal individuals and if there's a distinction between normal reference populations and disease populations. So in this analysis, we summarized measuring in two groups. The first group was a small group, about 500 individuals that were self-declared to not have any cardiovascular disease, were not being treated for cardiovascular disease, and were self-declared healthy. And then that analysis was followed up with over 3,000 individuals from the Framingham heart failure study, which were also self-declared as well as clinically identified to be healthy individuals that were not currently diagnosed with disease or being treated for disease. And you can see that in these two cohorts we get very, very similar numbers using our test. Now, in this case, you could be compromised using research use reagents because basically the median, based on our test, is the limit of detection of the research use test. So half of the normal population is effectively unmeasurable using research use tests. And there's several clinical trials that have used research use tests that have confirmed this observation. Further, we had to have some mechanism to define a threshold concentration that the FDA wanted for us to use as we went into clinical validation. Now, unlike a diagnostic test, again, which, the ideal diagnostic test would be elevated in patients with disease and would be very low or undetectable in patients without disease. So if you can detect it, then you would be positive for the disease. With a prognostic test, it's not so simple because you can have high risk in the presence of disease, low risk in the presence of disease, and you can actually have measurable concentrations of an analyte with no disease. And that's what's shown in this primary reference cohort, is that concentrations can range from very low single digit nanogram per mil. The minimum value that was seen was 2 nanograms per mil, all the way up to over 100 nanograms per mil. Now that's an individual that probably has pre-symptomatic disease, which we've learned since we did this, but again, these are self-declared healthy individuals. But the FDA wanted us to find a cut point that could be used to provide guidance to physicians in how to use the test. So we elected to use a strategy of defining that cut point based on higher concentrations in the normal asymptomatic population. So we picked approximately the 95th percentile, which in this analysis was shown at 37 nanograms per mil. Just for ease, we picked 35 nanograms per mil, and then used that concentration in various clinical trials to validate its performance as a cut point for prognosis in patients with heart failure. So moving into a definition now of the disease state that we needed to define the use of the biomarker in. If you're moving into a regulatory process, you're of course going to study the biomarker or your test in multiple disease states, multiple syndromes, but it's not reasonable and probably not even possible to get multiple clinical clearances. You really need to define a specific question and a specific objective in order to get a clinical clearance through the FDA or through any other regulatory body. So based on both our clinical evidence as well as the advice of our advisory board, the key opinion leaders and specialists in the area, it was understood and we pursued the utility of ST2 in managing patients with heart failure. So leveraging the prognostic power of ST2 as a heart failure management tool. So going back to, again, that first study, now using our validated test, we see that we get exactly the same profile, that ST2 concentrations are higher in patients that have diagnosed disease, heart failure, than in patients who have symptoms but do not have diagnosed disease. So again, the potential diagnostic but not a powerful diagnostic. Same profile of association with symptom severity, higher concentrations in acute heart failure, so worse symptoms and worse disease, you get higher concentrations, but you get the trend in both acute heart failure, which are hospitalized patients, as well as in chronic heart failure, which these patients are not hospitalized. This is a community cohort of patients that are being treated as outpatients with what is perceived as stable heart failure. Now in this case, you would also expect that a prognostic test would not be binary. Again, just because you're above a certain concentration doesn't mean you go from no risk to absolute risk. There's going to be some grades of risk as a function of concentration if your test or if the biomarker is performing appropriately. And that's exactly what we see with ST2, is that in very low concentrations, we have a very low risk profile. And once you cross the threshold, and this arrow indicates essentially that 35 nanogram per mil threshold, and then as we increase concentrations by decile, we get to very, very high risk, greater than a 50% risk of 1-year mortality at the highest concentrations. So it provides you something that's informative, potentially actionable, but certainly informative on what this patient's biological profile is in the context of their disease. Okay. Just like we saw with the initial Kaplanmeier curve, but now focusing to the left on that 30-day power, is that if ST2 concentrations are elevated or not, patients with a low ST2 that receive treatment have a very low risk of short-term mortality, but patients with elevated ST2, even in the presence of obtaining therapy, in this case standard therapy, not biomarker defined therapy, but standard therapy, still have a very high risk of mortality. So is this something that can be addressed? And that's an area that we need to explore further. Just like in the US population, this is a European population where we look at the risk of death or heart failure hospitalization. So another very meaningful endpoint in managing patients with heart failure is how frequently they get rehospitalized, because hospitalizations are both very expensive and very dangerous to the patients. And what we see in this case is that an elevated ST2 has a 17-fold higher risk of being either dying or being readmitted within 30 days than a low ST2. And that risk is persistent out to 1 year follow-up. And this was from a European cohort. So clinically, we do not see any differences in the biomarker between populations in different countries or different regions. So US population behaves the same as a European population, behaves the same as a Chinese population. So that, there's no ethnic or in this case regional, clinical differences between populations. So that's also very important in understanding how your biomarker or how your test performs. Now, a test also needs to be consistent. So this is a summary analysis from 3 different cohorts. These are all ambulatory heart failure cohorts, so they're well-managed outpatients. The first 2, the Barcelona and the PHFS, that's Penn Heart Failure Study, these are community cohorts. So these are all comers. These are patients who have a wide spread of different underlying etiologies to their disease. They're being managed, at different levels across a much broader age spectrum, et cetera. So they're very representative of the normal disease population, but a sicker population than by comparison to the HF Action group. HF Action was a randomized controlled trial, and in this case, the therapy that was being evaluated was exercise. So these are patients who do have heart failure. They are being managed for heart failure, but they were healthy enough to be able to participate in some form of physical activity, so an exercise level, as part of their therapy. So this is a much less sick cohort than the other 2. But what you see in both the left, which is a univariate analysis, you're only looking at the power of ST2 at the threshold concentration of 35 nanograms per mil to predict mortality in 1 year. The values are the same across all 3 cohorts. And if you look at the right panel, which is a fully risk-adjusted model, so it includes, I think, 11 different variables, the New York Heart Association class, age, gender, body mass index, renal function, et cetera. It attenuates the power of ST2 a little bit from a hazard ratio of over 4 to a hazard ratio of 3, which is normal. When you include more variables in your risk model, you are going to see an adjustment of the power of any specific variable. But the consistency across the 3 cohorts remains constant. And that's important, that you're not seeing variations in the utility or the function of a test from sources of your test subjects. It's very consistent. So that provides a lot of confidence and peace of mind to the clinicians and also to the regulatory agencies as they're evaluating the performance when this kind of consistency can be demonstrated. Okay, so taking that knowledge, we did go to the FDA, and some of the advice that I've tried to intersperse in this in this section was preparing to do that. So the first thing is to work closely with the FDA. The FDA will certainly have dialogue with you. They have a formal process to do that, but it is a process that is readily available. You can meet with them, you can ask questions. They won't tell you exactly what to do, but they will. If you make a suggestion that's just crazy, they'll give you some indication that it's a crazy idea. They won't come right out and say that, but they'll give you indications that you might want to try something different. And they'll give you some ideas of exactly what kind of things that they need. And then if you follow that, your probability, and your test can achieve it. So you follow their guidance and you can actually show the data to achieve what they're suggesting, your probability of a successful transit through the FDA is good. But you do need to know your analyte, you do need to know your test. All of the things that I just described are just a snapshot of what they're going to expect to see from your test performance, from the characteristics of your analyte, where it is in the disease you're interested in other syndromes and so on. You need to be able to answer those questions. And then have that clear and convincing evidence of the performance of the test and the utility of the test so that there's something actionable. Okay, because the FDA is focused on 2 things, primarily on safety. Is this test going to, if a decision is made on the test, is there a risk to the patient? So you have to be able to answer that question. You have to show that the risk is understood if there is risk and what the consequences will be. And then is it clinically useful? That's less of a question for the FDA. That's more of a question for CMS. But it's an important question for you as a developer of a product to show that not only do you get safety and effectiveness, but there's utility. So what would be the utility of a prognostic test? So as I mentioned, being able to establish the risk of an adverse event, whether it's mortality, hospitalization, worsening disease, whatever your definition of prognosis or of risk is, being able to define that is interesting, but unless you can do something about it, like for diagnosis, if you diagnose a disease, that defines how that patient is going to be treated. Prognosis is not so clear-cut typically. There are many different techniques. The Framingham Heart Score, for instance, is a technique to define in an asymptomatic individual what that person's risk is of developing cardiovascular disease in the next decade. But there's no clarity about what you would do about that. So if you're developing a laboratory test where somebody is going to pay money to perform the test, you're going to get a value. What are you going to do with that value to affect the future of this patient? So disease monitoring and therapy monitoring and treatment selection would be the obvious utilizations for a prognostic test. So is ST2 useful in that environment? So this is just one example from a randomized controlled trial. This is from the ValHeft study. ValHeft was an evaluation of a very large cohort, over 5,000 patients, for a drug called Valsartan and ARB for reducing morbidity and mortality in patients with chronic heart failure. Samples were collected at multiple time points. In this analysis, we're looking at measuring ST2 in patients at enrollment, so that's the baseline value, and then a second value collected 4 months later. And what we're looking at in this case is the risk in patients as a function of whether their ST2 value changed or didn't change over that 4-month period of time. So because this was a stable cohort, we would expect that most of the patients would have low ST2, and that's true. That there were over 1,100 patients that where it was the U.S. Subset of this cohort. It was a multinational trial. We only looked at the U.S. patients, and then those patients where specimens were available because they didn't collect blood samples from every patient in the study. So this was about 1,100 patients from this full 5,000 patient cohort. But they were stable ambulatory patients, so ST2 was low, and that's the low to low group. Now we have 3 other groups, of course. The persistently high group, so patients that had ST2 above 35 at both time points, or what we call lane changers, patients who were started out high and went low, above or below 35, or the converse, low to high. And what we see is that in a patient, we used the persistently low group as the reference, so their hazard ratio was set to 1, and then each of the other groups were compared to that. And we see that if a patient was persistently high, they're at much higher risk, 70% higher risk of an adverse event, mortality in this case. Or if they went from low to high, they were even higher risk, a 90% increased risk. But the patients who were persistently low or were high and went low had essentially the same level of risk. So we get the first snapshot here that monitoring the change in ST2 over time would potentially be useful. We don't have any information about therapy here. We only know from this analysis that the change is possibly useful. And that's been confirmed in now multiple analyses of serial testing and changing, is that a point measurement is valuable, but a change over time is even more representative of the patient's disease state and whether their disease is responding to therapy, potentially improving, or at least not getting worse, or if their disease is getting worse. Now, if you come to the business case analysis, one of the things that doctors are always concerned about is if you give them a new test, and they're already busy. Doctors, if we've been to a doctor's appointment, you usually get 15 to 20 minutes with your doctor, and they're booked every day. So if you give them a test that's going to recategorize their patients, is this going to fill up their waiting room with patients that are going to even exceed their capacity now? And in this case, we did the math, and it turns out no, that it actually helps them more effectively manage their patients, because you can very confidently identify those patients that are lower risk, which in this case is basically 80% of the patients were either in a low risk or moved to a low risk category, patients that could maybe monitored or managed more in infrequent visits. By contrast, again, we've redefined 20% of these patients in this very large population that are higher risk and probably justified having more aggressive follow-up pattern and more changes to their medication. So you actually can use the tool to more effectively manage your census of patients than they currently are through standard of care and symptom observation. Now, another analysis, this was done by a group at Mayo Medical Clinic, in a cohort of 200 patients where they were seen every 4 months for 2 years, looking at mortality as an endpoint, and evaluated the 3 top biomarkers for managing patients with heart failure, which is ST2, BNP, one of the natriuretic peptides, and cardiac troponin. And the question they were asking was whether serial testing, testing at each one of those time points, was a better utilization, or would measuring only one marker be more powerful. And what this shows is that the highest hazard ratio was achieved by doing serial testing of ST2 alone. So the most accurate risk assessment was with serial testing of ST2 alone. When other markers were added, when BNP was added, and then when troponin was added, the hazard ratio decreased, meaning that the accuracy of prediction decreased with the addition of the other data. So the conclusion from this type of analysis, and again, this comes to the business case, is that how would you recommend using the test? Well, BNP and troponin are outstanding tests for what they tell you about the patient's disease. BNP tells you about hemodynamic status, stretch, and volume. Troponin tells you about cardiomyocyte turnover and necrosis. They're great point measurement tests, but they're not as good for serial testing as ST2 is. So when you're going to a payment scenario, you're not telling the lab or the insurance companies that they need to measure each of these tests each time. Certain times they would measure all 3, and then certain times you would measure just one of them. So it gives you justification to go into those kind of financial discussions with the various entities. Now, of course, as you move into, heart failure is a very complex syndrome. Most of what I've talked about is focusing on later stage heart failure, stage C and D heart failure patients. So these are patients with clearly diagnosed, active, treated, and heart failure that's being treated. However, the earlier stages of disease, stage B, is that stage where somebody has had an event, and we're now moving into potential therapy and risk management. So in this case, ST2 was monitored, and the highest risk category was at 35 nanograms per mil, which is our threshold. So it occurs naturally within the population. And now you can use that to go forward. Okay? Another observation of change over time and potential therapy, and this is a cohort of patients that were hospitalized for heart failure. They were discharged, and then we looked at whether their ST2 changed during their hospitalization and whether they responded to a certain therapy. So if their ST2 level did not change, they had if their ST2 level did not decrease, they were classified as a non-responder and a high risk, and that's number 4. If their ST2 level decreased, they were classified as a responder, and that's category number 1. Now, in the outpatient environment, some of these patients received beta blocker, and some patients received other therapies. If they were a low risk, an ST2 responder, which was lines 1 and 3, there's no statistical difference. But if their ST2 level, if they were a non-responder, but they got therapy, the increased therapy as an outpatient, their risk profile changed. So what this tells us is that you can use the test to attenuate and modify risk, that it is actionable. It's not just an academic question. And those are the areas that we're now moving into for further clarification and adjudication. And I'll wrap up with just one case study analysis of a typical heart failure patient, an 85-year-old individual with mild symptomatic heart failure, very typical atrial fibrillation. But based on the standard heart failure biomarker, NT-proBNP, it's a stable patient. It's elevated, but it's not diagnostically elevated because this is a patient who has disease, so their BNP, their NT-proBNP should be at about that level. Over a series of visits, his NT-proBNP level is constant. No indication that this patient is at an increased risk over anybody else in the cohort. He's ambulatory, he's reporting feeling okay, and he's responding to his therapy, or at least tolerating his therapy. But when ST2 was measured, we see that his ST2 level is quite high, almost 80, and that's a very high concentration for a stable heart failure patient. So based on that observation, in this case, the doctor, who's an expert in biomarkers, started modulating his therapy, increasing beta blocker at various doses, and this was a patient who was already mildly hypotensive, had low blood pressure, so there was a concern that he may not tolerate the higher beta blocker, but he did. Add an increased dose of ACE inhibitor, got his ST2 level below 35, stabilized the patient. So now we see if we just use ST2, we get a very clear response profile over a series of visits. And this patient stabilized, tolerated the increased medications very well, reported feeling better, and a year later had no hospitalizations or adverse events. Okay. Now, some of the areas that, and I touched on this previously, was moving forward. So we established the ELISA as our first method. That's now being used around the world. Moving forward, we needed a more appropriate test for rapid turnaround time. Even though ST2 is a prognostic test, so short turnaround time is not an absolute requirement, but many laboratories don't like to do ELISA, so a test that's more appropriate to every hospital that provides a more reasonable 1 hour turnaround time was important. So we started evaluating technology that we could have access to that wasn't encumbered by other intellectual property, and that's lateral flow. So we have the reagents to do that, so we answered that question. We know the analytical use requirements. So could we develop a test to do that? We said we already have the reagents to do it. We know the form factor, we know where it's going to be used, how it's going to be used, and then could we develop a business case to do that, which we did work on. And now that project, that test has been, as I showed in the first slide, has been introduced under CE mark. We're now working on our FDA submission, and we're also already on the market with that test in most Asian countries and about to go on the market in China through their regulatory processes. So just one last slide on project management and how you would approach these types of projects. Now, if you're a Roche or an Abbott, you've got the resources to do this internally. If you're an earlier stage company, or in the case of Critical Diagnostics, a startup company to do this, you have to think about what do you do and what do you outsource. So we took the strategy of only doing the things ourselves where we add value. So fundamental research, fundamental product development, and certainly manufacturing are not things that we are good at. We don't have the resources to develop those processes, so we outsource all of that. Okay. And there's, in the area of immunoassays and diagnostics, there's many options to pursue that. There's capabilities for every step of the way. Very strong companies and research institutions that can do this. Whether those companies have the equipment and facilities, the capacity, whether they're aligned with your priorities, you have to decide those things. And then, who's going to handle QA and regulatory, which of course that's one of the areas that we take on because we are the manufacturer of record. So with that, I will conclude, and sorry that I did run over a little bit, but I'd be happy to try to answer any questions that have come up.
Thank you, Dr. Snider, for that informative presentation. However, since we are running short on time today, we will not be able to take any questions from the audience. I would like to say thank you and ask if do you have any final comments other than your conclusion?
Thank you, Christina. I don't have any additional comments. I think I interjected my recommendations and observations throughout the presentation. So again, hopefully this was helpful. It was my pleasure, and have a great day.
Excellent, thank you. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. And please share the announcement with your colleagues who may have not been able to attend today's live event. Thank you for now. Thank you for joining us. And we hope to see you again soon here at LabRoots. Goodbye.